next presenter is Associate Professor Lucy Sullivan, who actually was working here with us at the Alfred for quite some time as a senior researcher. She was also working at the Doherty Institute and is now currently the Scientific Director of Transplantation and Immunogenetic Services, Australian Red Cross Lifeblood, and Lab Manager at the South Australian Transplantation and Immunogenetic Service.
So thank you all for having me here today. Today I'm going to be talking to you about the association of non-HLA autoantibodies and clad following lung transplantation. Before I talk to you about a non-HLA, I'm going to be just introducing what is HLA. So HLA stands for human leukocyte antigens, and these HLA are proteins that are expressed in most cells in your body. And your immune system uses HLA to see which cells of your body belong to your body and which ones don't. And so if you have non-self HLA, such as in transplantation, this is called HLA mismatching. And the immune system recognises this and can trigger an immune response. And this immune response can either be a cellular response involving T cells, such as seen in this picture here, or an antibody response. And it can lead to rejection, either T-cell mediated rejection or antibody mediated rejection. Okay, so antibodies directed towards donor HLA are called donor-specific antibodies, or DSA, and the presence of DSA is strongly associated with rejection. So this is shown in this figure here. So on the y-axis is freedom from rejection, so if you're at the top you have no rejection, and time post-transplant on the x-axis. So recipients that have DSA unfortunately progress to rejection much faster than recipients without DSA. So antibody-mediated rejection happens in the presence of antibodies to HLA, but in a number of recipients, particularly in kidney and heart transplant recipients, they've noticed that DSA are not present in many patients who are diagnosed with antibody-mediated rejection. So this is up to 40% of recipients post-heart transplant have antibody-mediated rejection, but it doesn't appear to be driven by HLA molecules. So this prompted an investigation into the role of non-HLA antibodies in transplantation. So non-HLA molecules, the antibodies that develop towards them, these molecules function very, very differently. They tend to be molecules of what we call self. So these are proteins that make up either sort of the structure of your cells and tissues, or molecules that are involved in cellular functions. So unlike HLA antibodies, the development of non-HLA antibodies doesn't require a sensitising event. So HLA antibodies will develop either post-transplantation or following pregnancy or transfusion. And these antibodies aren't considered currently in donor selection for transplantation, but there is mounting evidence for a role for them in rejection following transplantation. So in terms of lung transplantation, what can happen is the underlying disease can cause damage, and this causes an increase in the presence of non-HLA antibodies. And following the transplantation, where you have further injury, these non-HLA antibodies can cause tissue destruction following transplantation. So this prompted us to ask, do non-HLA antibodies impact on outcomes following lung transplantation? So in our study, we had a cohort of 68 adult lung transplant recipients, and we looked retrospectively that we had stored sera on these recipients. And all of the recipients, we had clinical follow-ups with a mean time of 5.5 years. And how we looked at the non-HLA antibodies was using a commercial kit that's provided by a company called One Lambda. And this kit has 39 targets. on it. So we could look for the presence of antibodies to 39 different non-HLA molecules. And we looked both in pre-transplant samples and in a sample 12 months post-transplant. And we correlated the presence of these non-HLA antibodies to clinical outcomes, and most specifically chronic lung allograft dysfunction, or chronic, really it's a fancy way of saying chronic rejection. So the first results is we're now just looking at the number of different antibodies that the individual recipients had. So on the y-axis we have the number of recipients, and on the x-axis the total number of positive targets that particular recipient had. So as you can see, there was a broad range, and it ranged from some recipients having no antibodies positive on the panel, all the way up to a couple of recipients that had 18 positive targets. So now just looking at which of these targets were associated with chronic rejection, there were a few that were associated with chronic rejection if these antibodies were present prior to transplant. And that the names of the molecules aren't really important, but just to note that a couple of these antibodies had already been implicated in rejection following heart and kidney transplantation. There were also a few other antibodies to targets that were associated with rejection when the antibodies were detected 12 months post-transplant, and they're listed here. And once again, a few of those targets had been implicated in rejection following kidney transplantation. So the one thing I just wanted to point out was this molecule called CD36. This molecule is widely expressed, and particularly on platelets, which are involved in clotting. And we were the first study to pick up antibodies to the CD36 molecule post-transplantation, and it seemed to be associated with rejection if it was detected both pre and post-transplant. And interestingly, antibodies to CD36 have been implicated previously in transfusion-related acute lung injury. So this is a novel association that we're looking to follow up. So finally, this is a really important piece of data in that, so now if we just ignore which particular targets are positive and look at the number of positive targets in the recipient, and we had a real distinction between recipients that had less than 7 targets and recipients that had 8 or more targets positive. So recipients that had 8 or more positive targets had both a higher incidence of rejection and a shorter time to rejection. So this is sort of really important in terms of monitoring moving forward. So the take-home messages from this work is that we really do think there's a role for non-HLA antibodies in transplant outcome following a lung transplantation. But there is more work that's needed to define the specific targets. And one thing that we're really grappling with is sort of some more bigger questions, whether these antibodies are actually causing the damage or whether they're just indicative that something wrong is happening. So there's definitely more studies that are required to determine this, and particularly the mechanism that's involved in non-HLA antibodies and tissue injury. But we do think that testing for whether they're causative or just an indication that's a biomarker, if you like, that something's wrong, we do think that testing for non-HLA antibodies pre-transplant might be useful to help guide treatment to avoid antibody-mediated rejection. So this work was really driven by this very talented PhD student, Steve, who I believe is online. And this is how he comes to work every day. He's a super happy fellow and really enjoys his research. So thank you to everybody involved in the study, and particularly thank you to the Lungitude Foundation for supporting the study, and also a massive call out to the lung transplant recipients who kindly donate samples to the study. So without that, this wouldn't be possible. Thank you.